Instituto de Biología. Tengo que decirles algunas cosas antes de comenzar con la plática del día de hoy. Lo primero es este asunto de seguridad. Saben que tenemos dos salidas de emergencia, una por donde está entrando Ismael. Entonces los de esa parte del auditorio saldrían por aquel lado, los de esta parte del auditorio usando esta puerta. Esto en caso obviamente de alguna emergencia, ¿no? Alarmas, terremotos, cualquier cosa que pueda ocurrir durante la siguiente hora. Y bueno, para mí es un gusto tener a Michael Tesler el día de hoy. Es un gusto por muchos motivos. Uno, porque es acerca de sanguijuelas, ¿no? El grupo de animales que yo estudio. Y pues me da mucho gusto ver que otra persona está interesada en este grupo de animales. Otro, porque Michael es estudiante actualmente de Mark Sidal, que fue mi asesor de doctorado. Entonces estoy aquí presentándoles como a mi hermanito académico pequeño, ¿no? Y me da mucho gusto que esté aquí. Bueno, les voy a hablar un poco acerca de Michael Tesler. Él pues es un biólogo bastante particular. Tiene muchos intereses en la biología. Y una de las cosas que resulta muy interesante es que él empezó a estudiar, bueno, hizo una maestría en biología en Fordham University, donde estudiaba musgos. Entonces sus primeros años como investigador o como joven naturalista pues estudiando musgos, después cambió a sanguijuelas por el bien de nosotros, eso lo podemos discutir después. Y bueno, ha estado trabajando ahí, ahorita está haciendo el doctorado en esta institución que se llama, bueno, es un programa del Richard Gilder Grad School, que es en el American Museum of Natural History, esto es en la ciudad de Nueva York, y es un posgrado pues muy interesante, ¿no? Es bastante particular, son chicos muy talentosos, todos ellos, y que reciben mucho apoyo institucional. Entonces lo que Michael Tesler pues ha estado trabajando, tiene varias publicaciones, tiene cerca de 10 publicaciones, muchas de ellas con musgos, pero ya está también trabajando con las sanguijuelas. Y bueno, está aquí porque estamos trabajando con algunos proyectos, estuvimos en el campo, entonces no quería perder la oportunidad para que nos platicara un poco de su trabajo. Y bueno, entonces nos va a platicar y después tendremos la sesión de preguntas y después una sesión de café y galletas si quieren preguntarle más directamente, ¿no? Adelante, Michael.
Hey everyone, thanks for having me here. I really appreciate it. I'm very sorry that mi español es terrible, so I'm going to talk in English, so please be patient with me. I'm sorry to come to your country and then speak in English. But so I'm going to talk to you about some work that I'm doing on terrestrial leeches. So not all of you are probably aware of terrestrial leeches. Those of you who do field work in Southeast Asia, Southern Asia, Australia, if you go in the wet season, you're probably very aware of these guys. But here in the Americas, we don't have any blood-feeding terrestrial leeches, and we don't actually have any in this particular group that I'm going to talk about. So just to get you a little bit acquainted with the leech, this is a beautiful leech, Hemodipsa picta, and as you can see, you'd think by this being in focus that this would be the head. Most people think that leeches, the head is the fat part. It's not. This is the caudal sucker. They have a back sucker and a front sucker. This is the head completely out of focus. Okay, so just before I get started, of course, I'd like to thank, these are some people who have been integral in this work, although there are many others to thank, as well as the funding sources, the National Science Foundation and the New Yorkos Foundation. So again, you may not be aware of these leeches, but most leech bites are not like this. There are a couple that do go for particularly nasty places, but it's rare that a terrestrial leech will do this. Mostly you get them on your legs, sometimes other parts of your body. But, you know, so this is a quote from Haeckel in 1883, just to, people have been experiencing this when they travel, unpleasantly experiencing this in other parts of the world. One of the intolerable curses of this beautiful island, he's talking about Sri Lanka, I believe, of all its plagues, the worst, as I was afterwards to learn, by much suffering. So these leeches are all over the place where you find them. So if you've never seen them, so this is a leech in China that I was working with, just collecting. They kind of crawl around. They inchworm from one sucker to the next, and they're very common. But this guy's only about this big. It's pretty small. They're very small, but they can be very abundant. And so one of the things that people have been working on for a little while and was debated in the taxonomy for quite a while was how many groups really are there? And is this one family or are these distantly related groups? Nobody really knew. And so this is I was in Chile recently and I collected this guy. This is Untitled Chapter, one of the few blood-feeding terrestrial leeches in the Americas. And so as it turns out, there really are two clades, one Hemidipsidae, one which was resurrected as Xerobdellidae. And so this has a lot of non-blood feeders like Xerobdella, as well as Diastechostoma, which you can find. Mexico happens to be particularly speciose in Diastechostoma, but you wouldn't be aware of it because they don't feed on blood, so you have to put a lot of effort into actually collecting them. As well as Mesodella, which is the one blood feeder in the group. But Hemidipsidae is the one that I'm going to talk about today, which is the larger clade of blood-feeding leeches. So we didn't test the slides, of course, on this, so I apologize if some figures apparently don't show up. So this right here is a beautiful picture of the head of a leech. This is two. Right here, this is a three-jawed leech. This is a two-jawed leech. So there are two groups of leeches. One is proboscis feeding leeches. The other is jawed leeches. And so within jawed leeches, the ancestral condition or the plesiomorphic condition is to have three jaws. So you have kind of a jaw like that and two going like this. So if you get bit, you look at it under a microscope, it looks like a Mercedes-Benz symbol. Now, the other one is two-jawed leeches, and that's what I'm going to focus on for the first part of the talk. And the two-jawed leeches, this is the only group of leeches, of all leeches, that have only two jaws. There are other reductions in leeches, but never to simply two jaws. And this is the distribution. So three-jawed leeches, although a bit more speciose, are found mostly in southern Asia, a bit in Southeast Asia. And then the two-jawed clade is much more widely spread out, although this kind of extension over here is just one leech species in the Juan Fernandez Islands that kind of extends that a bit further. And so prior works found that the two-jawed leeches in red are really nested in a paraphyletic group of three-jawed leeches, which is mostly comprised of a single genus, Hemidipsa. This, at the time of this work, was debatable between whether there were somewhere between 12 genera and 31 genera. So one genus up to 31 genera. So pretty big disparity despite this being more speciose clade with a much longer branch. Okay, so to start, I'm kind of talking just about these two-jawed leeches, and we wanted to improve the phylogeny. So to improve the phylogeny, we basically wanted to add a bunch more samples. We added, in the prior work, they used 3 genes. We added a 4th gene onto this. And we did fairly straightforward analyses. We had 2 ribosomal genes, 18S, 28S, as well as the mitochondrial CO1 and CO3. We did maximum parsimony and maximum likelihood analyses. With parsimony, gaps were coded for the ribosomal genes, whereas amino acids were translated for the mitochondrial genes. So this was the first time that this has ever happened for me, but both of the analyses actually got the same tree. I've never really had the exact same topology, but these are branch lengths are for maximum likelihood that the parsimony result is identical in terms of topology. Full resolution, generally well-supported clades. But then we have a problem. And so these are subfamilies. So all these don't worry too much about what the names are, but just notice that it's very colorful, and the colors kind of pop up in a bunch of different places. Every single group, with one exception, is not monophyletic. Every group. So every subfamily, which is roughly equivalent to genera also in this group, is basically, from a phylogeneticist standpoint, complete garbage. And so when we're faced with something that's complete garbage, we have to kind of make a choice as to what to do with that garbage. How are we going to settle it? Are we going to deal with it now, or are we going to hold off on it? This was the second time this was very forcefully shown, and so we felt it was time to change it. So we sunk all the genera into a single genus. There were no characters that we were aware of to subdivide the groups further. All of the characters that were used up till this point for these genera were largely external, And they clearly, although consistent for a given species, clearly were highly mutable across the tree. And it has one incredibly strong synapomorphy, having 2 jaws, which, again, this is the only group of leeches to have 2 jaws. So we felt very confident in making this change. It also made the group quite a bit more comparable, as I said, in terms of number of species, as well as branch length to the other large group, Hemodipsa. The fact that this was broken into 30 genera largely had to do with one Australian worker who was very excited about describing new genera. And basically, if he found a species, it was a new genus. So he, out of about the 40 species in this group, there were 31 genera, which is a bit odd. But of course, Shootout Mexico Alejandro wasn't so happy when I told him about this. You know, he's on the Mexican-US border. He's, what are you doing, buddy? He's, you know, guns loaded. He's not happy with me about this decision. And so I just wanted to talk a little bit about making these decisions and why I feel like this was a good step forward and maybe encouraging people lightly to, when presented multiple times with the same problems, be bold, take a step forward, reclassify, because who else is going to do it if you're not going to? So again, you can take your tree like this, where you have this kind of wild Christmas tree lighting going on all over the place, and you can say, okay, well, let's wait till somebody takes the time, the taxonomy, to really hammer into this, divide it into several groups that are really good. So you could do that, or you can proceed and do something that you know makes sense, is phylogenetically justifiable, morphologically justifiable. And so waiting, there are a lot of good reasons to wait. One is poor support. So it happens that this tree is quite robust, but a lot of trees are, you know, we don't have a lot of support. So leeches, like I said, are divided into proboscis feeding leeches, so they have a proboscis, or they have these 3 jaws in general. And yet, we still use those classifications generally, even though none of our phylogenetic reconstructions have found them to be monophyletic, because all of the basal divergences in the phylogeny of leeches are poorly supported. So we're just not there yet. We can't really make an informed decision as to whether it doesn't make sense or it does, and how to split it up further. And so a reason for that could be that we don't have enough data. And so, you know, in the age of we're getting into more and more genomic level data sets, sometimes, I mean, I'm only presenting on 4 genes, but some people at least are getting into phylotranscriptomics, phylogenomics, and so we might get more data to resolve the tree better, and we might get a different answer. So it might be worth waiting. That's entirely possible. Or it could be hard to find synapomorphies. Like I said, for, you know, I personally at this level, and none of the prior leech taxonomists in literature had described any characters that make for, you know, maybe 1 good clade here and 2 good clades there. We don't have those characters, so they're just not present yet. And another fear is synonymy lists. So as practicing taxonomists, we're all keenly aware of synonymy lists and just how awful they are to drudge through the literature to make sure you really are fully going through all of the synonymy lists. So there's a lot to hold off. So gunslinging, you know, Alejandro probably does have some points. You know, hold up, don't be too bold all the time. But if you have strong evidence, you have a really good synapomorphy, like 2 jaws for this group, you have strong phylogenetic signal, you know, really good support, I think that, you know, it's probably worth making this step, particularly if you work on an odd group of organisms. So we study leeches. Alejandro, how many people in the world focus most of their attention on studying leeches? 10 people. So there are 10 people, 2 of them are in this room, right? And 3 of them happen to be on this paper. So 30% of the people working on leeches are on this paper. And the fact of the matter is, you know, most people, even in those 10 people, most of them don't work on this group. So when's the next time somebody's going to come along and actually take the time to revise the group? You know, this is a hard question to answer, but my guess is that nobody's going to do it for a long time at all, if ever. And so Alejandro and I have a bit of a different feeling about how higher taxonomy works. But I think we can all agree that higher taxa are not comparable. One genus is not comparable to another genus. So the genus I just kind of re-described, Chtonobdella, is not comparable in any way, shape, or form to the genus Homo. I think we all agree about that. So even in leeches, so here's my tree, here's a different tree, 31 to 12 genera right there, one genus. They're not comparable in terms of branch lengths, they're not comparable in terms of sister groupings. You know, nobody's actually, although there is a good bit of external variation here in terms of number of kind of rings per segment, so these are segmented worms. These guys have a lot of color variation that nobody's looked into. So we don't actually know that morphologically this group has a lot more disparity than this group. So higher taxa are not comparable. So although I could have spent the next year or 6 months going in trying to find a new character which may not even exist to break clades down further, I would suggest as practicing taxonomists, I think the time has come in molecular phylogenetics that we've gotten good enough at identifying high quality clades that we can come up with easy synapomorphies to move forward, because higher taxa are meant for communication only. They're not comparable. So I suggest that people spend their time more wisely, either on finding new species, on adding to their sampling for their phylogenies, and so on and so forth. So that was a special part of the talk just for my friend. Okay, so those data were really well polished. I'm going to get into some more phylogenetics that we're working on that you know, are really preliminary data that we're polishing to move forward, but mostly to talk about this clade Chtonobdella, the most speciose genus in the group. So I've been wandering around a bit in Cambodia here. I was in the fall, and then China collecting leeches last summer. To get more sampling, we've added about 60 new terminals, quite a bit more species. We've even added a few more two-jawed leeches. And again, same genes. We added another gene to the prior data set. And these are just very preliminary results, just using parsimony, although the likelihood I haven't done bootstrap support, basically you come up with similar answers. So this is roughly the large, and don't worry about any of the terminal names on these, but the tree is roughly similar to that prior published work I was showing you. So again, here's a beautiful picture of a two-jawed leech. So this is the clade that I was just talking about with two jaws. And when we go into this, so we're adding I only added 2 more species to this, 3 samples, 2 more species. So this is an interesting, was formerly Leobdella singularis, now it's Chtonobdella singularis. This is the only species in the group known so far that instead of biting externally is actually an endoparasite. It goes under the skin of Latoria species of frog in Papua New Guinea. So it's a pretty interesting thing. Here are other Papuan species described as Leobdella, clearly highly polyphyletic. So the more species we're sampling, we're just finding out more and more things are not right. So the kind of shift to just one genus is still making sense as far as I'm concerned. And here I added a single new specimen of the Manobdella, and again, it's forming a paraphyletic clade. Not quite as extreme as the other one, but again, just further evidence of that same sort of thing. Now here again, a beautiful picture of a 3-jawed leech. So this is the genus Chtonobdella, which I'm going to focus on now. So unlike the other phylogeny, although we're getting a lot of samples, we have many more samples for this than we do for the other phylogeny I showed you, we're getting almost no support in the base of the phylogeny. So we're really not able to resolve the full relationships of these with the data that we're using so far. But what we are able to do is really work through and find monophyletic species. So we're able to test the monophyly of species, which has been useful for the group. And so we're still adding, a lot of these are still undescribed species or yet to be identified. I really have to wade through some of the Chinese literature to get at what some of these species are. Oh, I'm sorry. But here, what you can see is we only have one case. We have one case. This is Hemodipsa sylvestris. This is the one I showed you crawling around on the ground in China, where we have it and it's polyphyletic. This is the most common species of all terrestrial leeches. We have no idea why this is going on. Although there are a number of groups that are pretty similar in external morphology, we still don't know what's happening there, and we have to go back and really review these specimens because this was a pretty odd finding. So now I'm going to get into how we're working on describing new species of leech in Chtonobdella, as well as potentially in the future for other groups of tiny leeches. So these guys are pretty tiny. The one I showed you on my hand was pretty big. That was Hemodipsa sylvestris again. That's a big leech. A lot of these guys are really small. And we know that there's a lot of potential diversity that we haven't tapped into yet. So putative species that have been included in the phylogeny but haven't been described by previous lab members. So one that we actually have quite a good sample of, which was kind of dubbed originally Philemon brown and now Chtonobdella brown, just a working name. Clearly monophyletic, very robust clade. This is what it looks like, and although externally it may be somewhat distinct from some other species, you can see it's got some patterning on the back as well as the sides. We don't have enough information yet to really work just based on coloration patterns, because the ethanol often leaches out the coloration patterns on old specimens, and this surely in life looked quite different. And this is about 2 millimeters wide, so it's a very narrow leech, and it's really hard to work with. So these are the general characters we work with. So here's external morphology. Largely, so they're segmented worms, so in each segment they have rings, and they're a predictable number through most of the body. So if this leech has 4 rings per group, we can tell probably what it is. But there are only so many characters here. There's often quite a bit more morphological difference in terms of internal anatomy, both the way that the organs are shaped for reproductive anatomy as well as their positioning. But again, we're working on really tiny specimens. So this is a Hemodipsa specimen I collected in China, and it's about 6 millimeters long and about 2 millimeters wide, maybe. And the specimen that I'm going to talk about is of this two-jawed leech is even narrower. And really these are about at the limit of what most of us can dissect. And we're not really able to get at the internal morphology of these guys. Somebody out there might be able to do it, but in our lab we've tried and we've botched a lot of specimens and really haven't been able to do it. So of course there have been solutions to this problem. People have been working on tinier organisms for a while now. This is not new. So this is Mycetobdella. This is in the fish-feeding leeches. This is a marine leech, but it's actually it's kind of interesting. It's on these mycet or opossum shrimp. It's thought to feed on them, although not conclusively proven. And here you can see they're beautiful histological sections of it. But there's some problems with histology. One is that when you have a tiny little leech and you chop it up into a bunch of little pieces, now you have a bunch of little pieces and you don't have a leech anymore. And really for a type specimen, we want the whole leech. We want to keep the leech. We don't really want segments of it. So although this is fine, we might be able to do a little bit better. And it's pretty time consuming, particularly if you want to make these post hoc 3D models of it, which are particularly useful for visualizing internal organs. So the solution that I'm going to talk about is CT scanning, which has been used for a long time, particularly in biology for organisms to look at their skeletal structure, to look at shells. And for those types of hard structures, it works incredibly well. And basically, the CT is it's an X-ray, and it just spins the specimen and continues to take X-rays. And with all of those rotated X-rays, you can then stack them together, and you can come up with with really excellent 3D reconstructions. But there's some problems. One of them is, for a long time, we haven't been able to get quite the resolution that we need to work on really small specimens. The other one is, obviously, I said, we're talking about things that have hard parts. Leeches don't have hard parts. Most invertebrates, or at least many invertebrates, not most, but all the soft-bodied invertebrates, they don't have anything to really contrast. But people have started to work on larger specimens, and generally for the purposes of kind of general looking at morphology, seeing how well CT works, people have started to look at things like, so here's a very large annelid, a large worm, very easy to dissect. But here is a virtual dissection of it using CT that's, you get 100% of what you would recover through the standard dissection, but you retain the whole specimen intact. And again, this is something you can dissect. Nobody really looked into really tiny things or not. And so for us, we wanted to work on really tiny things. And like those worms I just showed you, you need to both fix the specimen preliminarily. So we tend to, when we collect them in ethanol. It works generally well for dissections, and it keeps the DNA intact pretty well. Other common fixatives are AFA is commonly used, alcohol formalin acetic acid is commonly used for fixing various specimens in parasitology. And glutaraldehyde is really well known to be a great fixative for working on electron microscopy. So we tested 3 different primary fixatives, and then we stained / refixed with osmium tetroxide, as it's commonly used to increase contrast in electron microscopy. And then went through a series of dehydration steps before finally actually scanning. So this was the test study. This is on Microcomputed decora. So this is the North American medicinal leech, which Alejandro and Ricardo here are working very intensely on this particular group. And so, but they're easy for us to collect in North America. And we worked on juveniles to get a really small specimens because the actual adults can get pretty big. And here in 1 and 4, these are ethanol fixed. 2 and 5, this is AFA fixed. And 3 and 6, these are glutaraldehyde fixed. So the most obvious thing is that ethanol is not good. You're not getting nearly the contrast that you're getting with fixing an AFA or glutaraldehyde. And then the differences between glutaraldehyde and AFA are a bit more subtle. AFA does a really good job of showing one organ is different from another. So here we're looking at the brain, very clearly different from the surrounding tissue. Here you're looking at the brain. I can't quite tell that it's that different from the surrounding tissue. But in glutaraldehyde, you get much more resolution in terms of kind of the definition within an organ. You actually kind of see substructuring a lot better. But for the purposes of taxonomy, at least thus far working on leeches, we're kind of crude about it. We just want to see where things are in their general shape. So AFA seemed a bit better. And here we can recover where the neural ganglia are, as well as where male and female gonopores are. So these are hermaphroditic species that are male and female. And here is a 3D reconstruction of both the male and a female apparatus. And then here we were able to reconstruct the neural anatomy from the eye spots right here, going all the way back. And so that's something that would be incredibly difficult, if not impossible, to do with a standard dissection. So we were very pleased that we were able to do that. But we want to work on museum specimens. We've known about these undescribed species in the group for a while, like I said, from Australia, the one I've been showing you. And they're fixed already in ethanol because that's what we work with. So we wanted to try refixing an AFA, as we found that that had the best results for our purposes. And then to achieve the level of resolution that we felt we needed for describing the species and really being able to differentiate organs easily was about 5 microns, which is about at the limit of what our scanner can do practically for a leech of this size. Potentially you can get down to about 1 micron on our scanner potentially below that on some other scanners, but 5 is a fairly practical number. So we had to scan it in 4 different sections to achieve that. And again, this was a great way to do it without chopping up the whole specimen. So this specimen, this type specimen, is still 100% intact, which is very nice for future people if they want to scan it again when we get even better scanners that are getting sub-micron level resolution. So again, here's the species that I'm going to show you, which we were unable to dissect. Sorry. So these are the X-rays. So here are the eye spots. So these are just, one plane X-rays. So the eye spots, the jaws, the brain. Here you have mostly male reproductive organs. These are the epididymes. They're actually stacked one on top of another, which if you dissected it open, they're probably going to plop out, and that would be hard to tell, but we've never actually seen that in any other leech. And then the male and female pores for the genitalia, they're very clear where they are, particularly in 3D. You can trace out exactly where they come out, which although on many specimens of leech, it's incredibly easy to tell externally where these pores are. On a lot of specimens, you really kind of want to, ask your friend, hey, are you sure you agree with me that these are separated by kind of 4 rings? Because they're very important taxonomic parts, but if your specimen's kind of crunched a little bit at all, or even just sometimes on nice specimens, it can be very hard to tell. So it's very nice to see. Here we have gastric tissue, as well as more male reproductive organs, so these are the testisacs. And here is an inset, so this was kind of blown up from that corner, zoomed in. This is the nephridia, which is very, very tiny. And here's the ciliated apparatus on it. So these were details that we never expected to get, and they're very hard to find, but with a lot of looking, you can really dig into some additional anatomical parts that we're not looking at with most of our dissections. And here, this is the caudal end, this is the caudal sucker, and right here, this chamber with these 2 exits going out of it externally, those are what are called auricles on leeches. We don't 100% know what they do. It's thought that because these are terrestrial leeches, they put out a bit of lubricant for the back sucker to help make a proper seal. But we don't know, but nobody's ever actually even looked inside to see what these things look like, and yet it's very clearly this wonderful chamber that, who knows, that could vary between species. There may be interesting things to look at. So here is the 3D reconstruction of the external features. You can very clearly see the annulation pattern, so how many rings per segment, and there are clearly 4 of them. Although externally you should be able to see the male and female gonopores here, it's very difficult. Like I said, sometimes that's the case on specimens you're looking at under the dissecting scope, but here we can actually, when we look at internally, we can very clearly see the exact position where they come out. As well as this is the caudal sucker, the back sucker. It's important for this group to be able to count the number of they're called friction rays that are going around that. It's one of the few discrete characters in the group. And here it's actually been probably a little easier than the dissecting scope because you can rotate the object to kind of get right on that disc. So here's the part that's more exciting though, because obviously we could look at external features. These are 3D reconstructions of the internal anatomy. So here we have gastric tissue, and I'll walk you through a bit more later, but again, here are the male and female pores. You can see exactly where they come out, which is really nice. Here is this auricle in 3D, which again, nobody had dissected before. And the intestine of this happens to be highly convoluted, and that level of detail with a normal dissection, it would kind of all smush onto itself, and you really wouldn't be able to tell. You'd be able to tell that it was curved, but not that much detail. So we decided, or I should say a little bit more, my PhD advisor decided we should name this after his good friend who is a famous author, Amy Tan, and they've been in the field together, as well as she's in some of her books written about these leeches. But most people, you describe a leech and then for them, and they probably say, why did you do that? Leeches are terrible organisms. But she's kind of cool, and she actually said she was glad to be immortalized in this tiny little leech, and that she hopes to go to Australia soon and have about a dozen of them feeding on her legs. I think that's maybe a little ambitious. They're really annoying to have a dozen of them on you, but I'm glad she was very excited about it. But so here's the video rendering of it. So again, the head's the smaller part, the caudal end is over here. So anterior we have 5 pairs of eye spots. We have the digestive tract in gray. Here we have the 2 jaws, which are in this kind of purple color, as well as the brain going along here with the neural ganglia going back. Again in gray, digestive tract. Now we have male and then female reproductive parts. Again, we found the epididymes were kind of one in front of the other, which we hadn't seen before. And going back to the caudal end, we can see the rear part of the digestive tract, the red, those are testis sacs, so more reproductive organs. And here's the highly convoluted digestive tract, which I think there's actually more taxonomic information in the intestine right there than what we're seeing in prior classifications. And there's that auricle, that potentially lubricating organ. So don't worry about what this says particularly. Our new species is on the bottom here. Everything in blue is basically what matches it. So this happens to be quite distinct externally and internally as a leech. So it varies from all the other ones. These are other terrestrial leeches in the family that have 4 rings per segment. So if you want to find out more, that's terrible resolution, but I'm happy to send the paper. It just came out last month, which I was very excited about. So I'm happy to send that to anybody who's interested. But of course, we still have more things we're working on. Most of those species that I showed you in the phylogeny, we're not quite ready to process yet because we only have a single individual. And as we all know, there's, it's a bit sketchy to describe something based on one species unless it's very different. But this is one of the new species I added to the phylogeny of the 2-jawed leeches. Here, so this is a frog, this is a Latoria species, I believe, and the leech is under its skin right here. This is with the skin cut away. So you can actually have even several of these guys, and they're up to 1/3 of the body weight of these frogs, so they're pretty terrifying for the group. But the frogs, people catch them and they seem to be perfectly fine all the time. Nobody's catching these. I think the leeches have been caught free maybe 1 time, whereas people going to Papua New Guinea and catching Latoria frogs get these all the time. But it turns out this giant frog, or giant leech under there's also a tiny leech under some other species of Latoria. But it's too tiny again to dissect and really describe. So with Anna Phillips, I'm working at the Smithsonian, I'm working on describing this species. And again, these are really preliminary scans, but the 2D sections as well as 3D. And here's the leech in 3D. So again, the anterior end, the head is on the right, caudal sucker over here. It's a little bit bent because this was fixed in a frog. We didn't fix it ourselves. We have the eye spots and the jaws and the brain, the digestive tract. And again, I showed before, I'll go back to that in a second. But here, those are the epididymes. They're off to the sides. They're not stacked one on top of each other. These are quite normal for the group. And back to the intestine, you can see this is actually very straightforward, the intestine here. It's very clear, it's not convoluted at all, which is quite different than the species we were working on. So just to wrap up a bit, we revised the higher taxa in this group, particularly the 2-jawed leeches. We sunk them into a single genus. I would recommend for the other molecular phylogeneticists out here, even if you're not completely a taxonomist, maybe be a bit more bold with reclassifying, and certainly consider, is anybody else going to do it in the future? The other thing is that the micro-CT appears to be really excellent for describing small leeches, and it's probably great for describing new large species of leech as well. The other cool thing is that the data are freely available, so it's kind of like the gen bank of morphology. Anybody can go, get the original scans, and they can pop it into their computer, and they can retrace out all the organs they want, say, hey, no, you mistraced something, or we found this new character. So those data are freely available for this type, so nobody has to come to the museum or if they're not a large institution, which would have a hard time getting a loan, they can just go on their computer and get it. They need a good computer to work on it, but they can get it. And this does appear to be the first soft-bodied invertebrate species to be described primarily using micro-CT scanning, although Fernando Carballo, as well as in a paper with Gonzalo Giribet, is also publishing the first flatworm to be described primarily using CT, so they're coming out in the print version at the same time, so they're kind of co-first soft-bodied invertebrate. So it's exciting that people are starting to advance these methods pretty rapidly. And so with that, thank you all for having me. Thank you for tolerating my English, and if you have any questions.
Bueno, si tenemos tiempo para algunas preguntas. Alejandra.
I love the scans, and I have two questions. First, how long did it really take you to get those images at the beginning?
Yeah, so this one was quite difficult. For one, I was new to CT. I'm more of a microscope kind of guy in general. But these took a long time to get. So one of the problems was, this was kind of early on in working with this, and it was actually scanned in 4 sections. And although generally that's easy to stitch together into one working part, we weren't able to do that for this scan. They were just a little bit too off. So I had to work on each segment individually. So if an organ overlapped, it was quite a pain to work on. But we've gotten better at doing it, and now we can always make it so they stitch together. So now that I'm good at doing this one took me probably weeks to a month of computer time. I mean, let alone the staining and scanning. But those are relatively simple. But probably a month of sitting down at the computer, clicking, scrolling through, tracing things out. The new one that I showed you from the frogs, it's not complete, but that was 7 hours. So I've gotten good at it, I guess. The other thing is that I do feel like, although I'm showing you pretty images and that's kind of how I want to razzle-dazzle you, actually the CT is great because you can get the 2D images. And for describing species, they're probably enough. So if you want to do rapid descriptions, you can very easily stain a whole bunch of things in a row, pop them under a scanner. It's extremely minimal effort. You go in, you look at the 2D scans, and you can manipulate it so you get the exact right plane of view for your scan, and you can see all the features you need. And so that you can do for a description if you wanted. You could do it in, get at each one of your parts in an hour, probably. So I would say probably comparable 1 to a few hours, same as probably it takes most of us to do a normal each dissection.
So is that the new scan they have at the museum, or is that the old scan they have at the museum, the CT scan, or is it a special?
No, it's the same CT scan. Yeah. And actually, people are starting to advance these methods on plants as well. So there was a paper that came out a few years ago. It was pretty cool, testing a bunch of the methods. They haven't, I don't think people have done small plants yet particularly, but
yeah, we were trying to do meristems with the CT scan at Cornell University, and we're still having a lot of problems. That's why.
Yeah, so I mean, it's working well for us, but yeah, plants are a whole different ballgame, and looking at what I've seen of some of the CT scans of plants, I'm like, oh, that's nice, but maybe not quite there yet.
¿Alguien más?
How strong is the X-ray? Like, I was thinking in seeds, in plant seeds. Like,
What's that?
I was thinking in doing a study on plant embryos.
Okay.
But I would like to germinate them after the analysis. Is that possible?
No, because of you have to fix them and stain them, and so you'd have to fix them. Otherwise, it would be great if you could do live tissue, but unfortunately, no. You'd have to just grow and randomly pick from your sample and do it that way.
But like in humans, when you get a scan, you get this contrasting compounds.
Yeah.
They don't kill you, so has anybody tried to do?
Yeah, no, that's a great question. And I'm not 100% sure. I think the resolution that they're working with is not quite the same that we need for organisms of this size, or you know, just barely germinating plants. But yeah, that's something to look into. But I believe the contrasting agents just aren't quite strong enough. Yeah, for this level of detail.
Do you happen to know what is the effect of these other fixating agents in the DNA?
Oh, the DNA's gone. Yeah.
Sorry.
Yeah.
It's gone.
Yeah. When I said it's non-destructive, that was maybe a bit bold. You know, like fixing your fish in formalin's destructive. So anytime you stain or fix in something else, I mean, AFA is partially formalin. So, you know, we're destroying that part of it. Yeah, so you're not getting DNA out of this, but the actual specimen is there. It can be rescanned, it can be redone. You can look at the leach as a whole. So it's more that it's less destructive. It's maybe you know, as putting in a leach in ethanol is actually destructive. Anything you do is mildly destructive. This is somewhere in the middle.
But you treated these specimens first in ethanol and then you changed to these other agents, right?
Yeah, so this is a museum collection. So this was just collected 10 years ago. It was in the collection. We knew molecularly that it was distinct. But we couldn't get at the internal anatomy, so it wasn't until people started getting better and better at doing CT on soft-bodied organisms, and that we were able to crank out even more and more fine-scale resolution, so down to 5 microns on the machine, that it became viable to do this type of work.
No, más preguntas. Bueno, muchas gracias.
Thank you.